So I feel like a lot of it. Do something.
You must have a very weak battery. Or something. I mean, either that or you forget to charge it up periodically or whatever.
This is the only time it gets plugged in. Things that I just forget at night. But if it bothers you, I can.
Oh no, no, it's not unusual to have at least one person plugged in, if not more, because there are 4 outlets. So.
See, I like the other room. That's
That's
what you
the
should have.
That's the what?
The outlets are in the table. In the other room.
Oh, well, there's some down there too.
Yeah, but no,
They're-
each table.
Oh,
in
I
the
got you.
room-
Okay, the ones that are in the table. Yeah. Okay. Yeah. Pardon me? Well, eventually these are supposed to be replaced out too to have the outlets in them. But the problem is that in order to do that, they have to core the floor multiple times and then put in, new circuitry in the walls and things like that. So it ends up being pretty pricey to do that. So, what's that? I guess it's a relative term, right? And also can be very loud and noisy when they're doing it in the summertime. Okay, so let's get started and finish up with our B cells and antibody production and go on to more cellular immune aspects. This painting, which is part of the Wellcome collection, which is the largest charitable organization in Great Britain, essentially is the foundation supported by the Wellcome Drug Company. And so they have a very large collection of medically oriented paintings and prints and things of that sort. And this one by Hielmacher is of Edward Jenner vaccinating a boy, kind of in a very romanticized version of that scene. Of course, he had no idea what actually happened. But this is an example of a genre painting. And in fact, it's a genre painting, because genre as a noun is just a class or a type of painting. And in academic art, there's a whole series of types. So you have landscape art, you have portraiture, you have still life, you have genre paintings. And depending on which one of those genres you had, it was, maybe not as respectable as some others in the academic realm. So genre paintings are basically everyday painting. So the scene is just something that would occur most anywhere. Of course, this maybe is a little bit historical. That's another genre. And realism with romanticism elements within it in terms of the style here. And of course, this is a very unique situation in terms of challenging our immune system. Because what we're trying to do here is protect people against smallpox. But what we're going to do is actually introduce cowpox into the person. And it's just because the antigenic determinants in the cowpox are so similar to smallpox that you get basically protection, cross-reactivity between the antibodies that are generated against the cowpox antigens, which can neutralize smallpox as well. So it's I don't know of another example of a vaccine in which this kind of antigen cross coverage is really used. Almost all of the vaccines are either the exact killed organism or weakened organism or a protein from that organism. We don't use a protein or an organism from something analogous to it, except that this particular example. And as it turned out, it was certainly a very fortunate occurrence that there was this similarity in antigens, because normally, again, we don't fool the immune system so well that it would produce a strong vaccination response against a slightly different organism. So one last time on here with this diagram, and before, I've always talked about that as just the antibody on the surface, okay? But we saw last time that this IgM in the immature B cell is actually the B cell receptor component, right? So that's IgM, and then we have these accessory proteins, shall we say, that provides the complete receptor. The IgM provides the binding specificity, but it has no intracellular domain. So in order to get an activation signal through, we use these associated proteins that are in, again, close association with the IgM. And so when antigen binds, then we're going to get these other proteins transmitting the signal into the cell. And the signal that we're going to generate is associated with phosphorylation of proteins. Very common mechanism that our cells use to transfer or generate signals from receptors. For B cell activation, the antigen binds to our receptor. And remember, they cluster. We have these antigens. These are, generally pathogens with hundreds of thousands of these antigenic molecules on their surface. And so they'll be able to bind multiple B cell receptors and cluster them. And that by itself might be able to activate the B cell. But many times we also need a co-receptor to be activated as well, okay? And also, as we saw last time, T helper cell help, okay? So those multiple components that we've already talked about need to go into the triggering this activation cascade. And now we're just taking a look at the inside of the cell and adding a little bit of the details with regard to antigen binding, generating a signal which will generate a proliferative signal, differentiation signals, things of that sort. So here from that prior dot, here's our IgM, and here is just two of those associated proteins. They didn't put it on the other side just to try and simplify the diagram a little bit here. Okay, so we get, and for the time being, forget about this stuff over here. Okay, it's just the antigen. So antigen binding the IgM on the receptor now triggers these proteins adjacent to the IgM to change conformation. And now there's a protein within the cell. Yes, there is. Okay, this guy over here is activated by these conformational changes resulting from antigen binding. And that protein called LIN is a protein kinase. It phosphorylates protein. And the protein that it phosphorylates is the insert in the inner domain of these proteins associated with the IgM. So those phosphates, those P's on there, were put there by LIN. Okay? And when that happens, a protein that is not normally bound to the receptor, called Syk, binds to this phosphorylated inner surface, inner proteins, and it also gets phosphorylated by LIN. Okay? Now, once that happens, for some antigens, that's sufficient to generate an activation signal, and there'll be a whole series of additional proteins that become activated, and they'll be basically will come into this pathway here with some other using other proteins. Okay, so Syk is called an adapter protein. It basically is present in the cytosol, and it's ready to bind to the appropriately phosphorylated protein, which is invariably a receptor. Now, for some antigens, for actually a fair number of antigens, that's not enough. So this process on the left-hand side isn't enough to get that B cell fully activated so it doesn't undergo apoptosis. What it needs is help from complement. Okay? And specifically the C3D, so one of those cleavage products from C3, right? That was one of the first interconversions from C3. We had C3b, C3d, C3a, that were products from that. Well, if this antigen is able to bind complement, or complement recognizes, again, it's a pathogen most likely, then we get the activation of the complement cascade, and generation of C3D, and that can bind to antigen. And then we have a receptor on these immature B cells, originally called CR2 for complement receptor 2, now called CD21, that can bind. It's next to the B cell receptor. It's pretty close to it, so it can bind. And then again, it doesn't have much of an intracellular tail. So it can't really do too much in terms of transmitting a signal. So we have CD19, which is adjacent to the CD21, and that's the one that is going to, with the change in conformation, that there is another protein kinase, not shown in this particular case, that phosphorylates it. And now we get some other adapter proteins, in this case called VAV, that will bind, and then subsequently we'll start to see the complete activation signal that we need. Actin polymerization, that's going to have something to do with the cell structure and then the ability to proliferate. And then this PI3 kinase, that's a second messenger generator that'll provide this extra signaling that's needed in order to get complete activation. Okay? So, B cell receptor is IgM plus some associated protein. The co-receptor is CD19, CD21, and CD81. CD81 seems to be a scaffolding protein. It basically is there to hold everything together with regard to the co-receptor. So it doesn't have any direct effect in terms of the signal transduction process. So this engagement of the co-receptor is seen for a fair number of antigens, let's put it that way. So it's not an infrequent occurrence. And what we can do sometimes in terms of if we want to modulate the B cell and its production of antibodies, we can do things to the co-receptor, disrupt it one way or another, and block antibody production. Okay? Because so many antigens are dependent on the co-receptor for that. So, in summary, B cells are going to bind antigen via the B cell receptor when it's been expressed in the immature B cell. Where that begins, it's the IgM that is characteristic of the immature B cell, that is the critical part of the B cell receptor that engages antigen. And then, depending on what that antigen is, we may need to engage a co-receptor to get activation of the cells. And again, this process is where we're looking for clonal selection and then somatic hypermutation to get some daughter cell that is producing now an IgM that has a higher affinity for the antigen. And we'll go through this cyclical process before we end up with a mature B cell. We may need T cell help for this in addition to that, although it's not essential for a certain pool of antigens. It's that we need the T cell help, just like there's only a certain pool of antigens that we need the co-receptor help. Okay, but for some you need all three: receptor binding, co-receptor interaction, and T cell help. Yeah.
Does the T cell help the CD40/CD154
Yep. Uh-huh. Right. So from this, you get the idea that not all antigens are the same. Okay. And so there are those that are so-called thymus dependent. So those are the ones that are going to need T help. And then there are thymus independent antigens. And the thymus independent antigens, in some respects, you can think of them as resulting from much older interactions between our immune system and these pathogens. And so they've become almost independent of some of this requirement for selection and things of that sort, because our immune system is already primed for these kinds of antigens. So, oh, well, how about a question, though, before we get into that? So, which component of the B cell co-receptor is phosphorylated upon C3b binding? Is it the CD81, the 19, 21, or 40? That co-receptor has 3 proteins associated with it. Only one gets phosphorylated. So, CD21 is going to be the portion that doesn't have an intracellular tail, so it couldn't be phosphorylated, and interacts with your C3d. CD19 is the protein that provides the intracellular piece that is required for generating a signal. And CD81 is, again, structural. It's there just to basically hold the 19 and 21 in the right location near the B cell receptor. Okay? So it's 19. There's the 2 phosphorylated groups, which in this case are almost always tyrosine. Okay, so T cell independent TI antigens. Essentially, there are a small group of individuals who are born without thymus or have had for some reason, their thymus has developed, and yet they're still able to raise antibodies. So clearly they don't need T help because you don't have the thymus in order to mature the T lymphocytes. So they're able, so against these antigens, you can generate an antibody. These particular antigens are divided into 2. There's the TI1 and the TI2 antigens. And they really divide out into the TI1s, so the thymus independent antigens, will stimulate both immature and mature B cells. Okay? So remember, so these guys haven't undergone the affinity maturation of the antibodies. So if you stimulate them, you're going to get all sorts of antibodies. These are the mature ones, are the ones that have undergone affinity maturation, and they'll give a monoclonal response. Just one cell that has been stimulated to produce antibody. So for TI1 antigen, because both of them are being activated, you'll get a polyclonal activation of the immune system. So you'll basically get these people will have high levels of circulating antibody, but it's not particularly specific against anything. You might see, some antibody clearly will be against the antigen that's being, is being challenged, but there'll be others in the pool as well. And so one of these antigens is called LPS, lipopolysaccharide, that's from the cell wall of gram-negative organisms. And it, again, just seems to be an antigen that our immune system over the years has somehow recognized as a so-called mitogen, which is simply a compound that stimulates cell proliferation. You do need a fair amount of LPS in order to do it. So this would be a person that would have, for example, a very extensive gram-negative infection will start to produce high levels of LPS and get this polyclonal activation. For TI2 antigens, these are only going to activate mature B cells. They tend to be carbohydrates or proteins that are in high density on bacterial cell walls. And when the immune system is exposed to these, you'll get essentially mature B cells starting to proliferate. Again, it's not uniquely as specific, so it'll be more polyclonal. You'll get these cells. In this case, there's a certain population of B cells, called B1 cells, that seem to be driven by these TI2 antigens. And the importance of it is that our immune system doesn't really develop a lot of B1 cells until about age 5 or 6. So little kids are particularly sensitive to certain types of polysaccharide antigens because they have not developed these cells that respond very aggressively to these types of antigens. Okay, so they're more susceptible to infections with them. So those are thymus-independent antigens, and then thymus-dependent, or so-called TD, or antigens, are basically everything else. They require help. They require T-cell help, and these are clearly just some exemplars. Diphtheria toxin, viral hemagglutinin, a purified protein from tuberculosis mycobacterium. So just, almost every kind of category, be it virus, bacteria, will have thymus-dependent antigens that we respond to. So this table just kind of shows some of the characteristics in terms of each of these antigens, the thymus-dependent antigens. This is what we've essentially been talking about in terms of, so will infants respond? Sure, they will. Are we talking about production in athymic individuals? Well, no, because they're TD, they're thymus-dependent. Antibody response and absence of cognate? No, because again, they're thymus-dependent. Activates T cells? Yes. Polyclonal B cell activation? No. Requires repeated epitopes? No. Induces immunological memory? Yes. Right? So whenever we get these antigens requiring help and going through the whole process of clonal selections, hypermutation, affinity maturation, basically, we're going to generate memory cells, memory B cells. In contrast, the TI1 antigens, again, infants will respond to them. Again, it's the TI2s that we need the B1 cells specifically to respond, and we're not going to see that until at least age 4, 5, 6, something like that. Antibody produced in athymic individuals, sure, because these are TI antigens. Antibody response in absence of cognate, absolutely, TI antigens. Activates T cells, well, no. That's the whole point. Polyclonal activation of B cells, the only one that does that is the TI1 antigen. Requires repeated epitopes for the TI2s, and that's what activates the B1 cells. And then induces immunological memory, no, because we don't go through the process of taking an immature cell, maturing it, and getting this monoclonal production of antibodies. That is, the memory cell development is part of that process. So for TI1 antigen, I already talked about LPS, bacterial lipopolysaccharide. Brucella also has antigen, that pathogen that will respond to in a TI1 response. And then for TI2 response, pneumococcal polysaccharide, salmonella. So those two, again, for little kids, they're a little bit more susceptible to, well, they're a lot more susceptible to because of the lack of this ability to respond to that type of antigen. And just to, I guess, reinforce, that's a good way, back to our B cells that are thymus dependent and are going to go from a resting B cell. And so the resting B cell and the immature B cell is the same thing, okay? So do they have surface immunoglobulin? Yes, they have IgM because that's part of the B cell receptor that they're going to be using to recognize antigens with. They have MHC class II on their surface. Do they make a lot of or secrete a lot of immunoglobulin? No. At this stage, all they're doing is making immunoglobulin that's going to go for the B cell receptor, and then as it transitions into the mature, we also get IgD on the surface, but it's not secreted. Is it inducible to grow? Sure. Somatic hyperinfiltration? Absolutely. Isotype switching? Yes. What's another good word for it? Absolutely. Positively. Positively. Okay. Plasma cells. The end-stage terminally differentiated cell. Surface immunoglobulin. No, remember that the high affinity receptors get stripped off the surface of plasma cells because they don't care at this point in time, they're terminally differentiated. They're going to make this antibody that they have affinity matured, and that's it. They're not going to respond to any antigen. So immunoglobulin, no, we don't need B cell receptors. Surface MHC class II, no. High rate of immunoglobulin secretion, yes, that's their sole purpose in life. And then inducible, no across the board because, again, they're terminally differentiated. You're not about to change them in some way from what they've already undergone. So a question about TD antigens, thymus-dependent antigens. Which of the following steps is not required for antibody production? These, remember, are thymus dependent. So we have to have interaction with T cells. Polyclonal B cell activation means multiple cells are being activated irrespective of their antigenic specificity or selectivity. Okay, and so things again like LPS are able to induce that. They basically just trigger the cell proliferation. So we don't see polyclonal B cell activation except in that unique situation. Naturally, I mean, there are some pathologic conditions where we'll get polyclonal B cell activation, but and so when we get antigen, the thymus-dependent antigen binding to the B cell receptor, that IgM, we get phosphorylation of Ig alpha and beta by Lin. Okay, that's part of that activation process. We get B cell antigen processing in order to express the antigen in MHC class II for interaction with T cells. And then, of course, we get affinity maturation, that whole series of, again, clonal selection, everything like that. So, but this is a unique situation, polyclonal B cell activation, for just, at least naturally occurring, for just a few antigens that are able to drive immature and mature B cells. Okay, so secondary immune response. The first response, the primary immune response, is going to set in place these memory B cells, okay? In addition to that initial production of IgM followed by many times switching to IgG, or it could switch to IgE or something of that sort. But basically, that's our primary response. It takes 1 to 2 weeks before you're going to see significant amount of antibody produced, but it'll come up, and that single exposure might be sufficient in order to produce enough antibody, enough memory B cells to handle that particular antigen. Notice that we produce a lot of memory B cells. They are for a particular activated B cell, a fair number, but in the overall context of B cells, they represent a small percentage of the total B cells that we have in the body. And when we get that another exposure, it's these memory cells that are going to become activated. And we don't want another primary immune response for most circumstances. Depending on what is the pathogen and the like, we might start a primary response on a totally different antigen. But with regard to most pathogens, we just want to be able to reactivate or activate our memory cells to start producing antibodies quickly. So, as I said, you don't want to start the process all over again with another primary response. And so what happens to prevent that is that with exposure to the antigen, these memory cells, which again, they have a high affinity B cell receptor so that they will recognize low levels of antigen exposure and start to proliferate and produce antibody within a short period of time. Within a couple days, you'll start to get antibody production. That antibody will has the capacity to bind to the antigen on the second exposure to the pathogen, and with it, we get the complex, so the antigen plus the antibody binding to naive B cells, which express the Fc receptor for antibody. So these receptors are IgM, so the mu constant region is what they recognize on these B cells. And when that's occupied, they will essentially inhibit the activation of that naive B cell. Memory cells don't have these Fc receptors on their expression, so they're not impacted by this. Okay? So this is the way that we can keep our primary activation reduced on secondary exposure to an antigen. Those initial memory B cells will have high affinity receptors on their surface because they were produced in that first round of that first exposure and the round of clonal selection and somatic hypermutation. As it turns out, the memory B cell, when it gets exposed to antigen again, will start and go through at least one round, maybe two, of somatic hypermutation again. So its affinity may in fact be enhanced in this process. And again, the receptor, the IgM there, is the same one that is being produced by the plasma cells from these stimulated B cells. So what we can end up having is antibody on the secondary exposure having higher affinity for the antigen than in the primary exposure. So sometimes that's also a reason for when you have a vaccination and you have to have multiple exposures. It's not only that you get a more pronounced production of immunoglobulin and also production of memory cells, but you actually get a little bit better affinity antibody, okay? So these memory B cells, they'll have, again, they already have the appropriate B cell receptor, but they do have MHC class II, and in terms of the need, because again, they're going to have key help in terms of driving them through proliferation to the final plasma cells. They tend to have a little bit higher surface levels of these molecules, so that, again, they're more robust in terms of differentiating and proliferating. So several different things that make the memory cell response more robust than the initial one. And this is shown in this diagram here. In the upper one, so down on the bottom, you're looking at an immunization protocol in weeks. And so you first have your initial primary exposure. 4 weeks later, a month later, you have a secondary one, and a couple weeks later, you have a tertiary one. So if we look at the IgM and the IgG, remember that IgM comes up first in the primary response, and then we'll get isotype switching for IgG, and as we get the second and tertiary response, we bump up the amount as all of these memory cells are moving on to plasma cells and producing antibody, and so we get a significant amount of antibody produced relative to that first exposure of antigen. At the same time, if we look at the affinity, with each secondary and tertiary exposure, we get increased affinity. And again, that's that 1 to 2 rounds of hypermutations of the immunoglobulin gene that enhances the interaction between the antigen and the antibody. Okay, and so both increase as a function of multiple exposures. So in terms of antibody production, right, B cells, but we need T cells, T cell help. B cell activation produces phosphorylation on the internal side of the receptor within the cytosol, and many times we need activation of the co-receptor in order to get complete activation of the cell so that it goes on to affinity maturation and possibly plasma cell, depending on if that's the cell that has the highest affinity for the antigen. And the second response is faster because you already have these memory cells with high affinity B cell receptors on their surface. So as soon as they see a small amount of antigen, they start to respond and you get antibody production within a few days. And then again, they'll go under hypermutation and increase the affinity from the initial affinity of the antibody. Okay. So that's it for the time being for antibody. Any questions about this? Yeah.
This is kind of over question.
Uh-huh.
Will this information, like specifically from slide 711, the B cell activation, is that going to be on our exam on day after tomorrow?
No.
Okay. So I'm probably going to eat your head on this one.
Okay.
At the end of this one.
So it finishes with a B and T cell interaction, which is the one just before. Yep. Okay, I think. Let's just spend a couple minutes with looking at T cells. And specifically, well, we'll go here. So as I said before, because T cells are provide so much help for antibody, we kind of had to talk about at least one type of T cell and not the whole, a lot about T cells, just to get through the antibody stuff. Okay, so now we're going to take a look at T cells as their own topic. And up until 30 years ago, there wasn't much known about T cells because, frankly, we didn't know or couldn't differentiate the different types of T cells. They were just a bunch of cells there that kind of looked alike, but they seemed to have something to do with intracellular pathogens. And so any microorganism that was able to enter a cell and in some way alter its metabolism seemed to be able to attract these cells, and these cells were able to kill, for example, virally infected cells, things of that sort. And so they were felt to be just tissue specific or tissue localizing cells of the immune system. And of course, now we know that they have a lot to do with the immune system in general and antibody production and regulation. And so what we're talking about in terms of the T cells, so this is just the different types of circulating cells. We have over here monocytes and neutrophils and basophils and eosinophils, but over here is our lymphocyte line. So that's the other major class of circulating cells, lymphocytes, about 20%, something like that, of the white cells that are circulating. And the common lymphoid progenitor cell can be initially segregated into lymphoid progenitor cells, and so these are the ones that are going to become T cells. The progenitor B cells, those for the most part go into the spleen and lymph nodes, and in that location they do their thing. And then we have this other cell, the NK cell. Remember, that's that innate immune kind of cell that has attributes of T cells, but doesn't have the specificity, but it does like to kill virally infected cells. And then once we have this lymphoid progenitor cell that's going to go to the thymus, we then go through a series of steps to differentiate the helper cell population from the cytotoxic population. So in terms of effector cells in the T cell lineage, we have the cytotoxic cells. Those are the guys that go to the tissue and can kill cells directly that have something like a virus in them. Helper cells, the TH4, CD4 cells, the TH cells, and then those helper cells, it turns out, are of two types. One that help make antibody, and one that helps monocyte macrophages. Okay? So what we'll do on Friday is take a look at how we get from this cell up here to these cells down here. Okay? So just a preview of things to come.